Today I'm going to go over topic 1 for this week. This is going to be microbial cell structure. So this goes over chapters 1 through 3 in the book. However, it doesn't go over each of the sections within those chapters. It only goes over select sections as you will need for this class. The topics for this lesson is to explain the general role of microbes from the human perspective, describe the properties of different members of the microbial world, examine the characteristics of organic molecules found in cells, and describe the bacterial cell structure. So in chapter 1, we're going to be looking at humans and the microbial world. So first of all, just to see how universal microbes are, your body carries so much bacteria that it's been seen to be anywhere from a 1-to-1 ratio of your cells to bacterial cells, all the way up to 10 bacterial cells for each of your 1 cells. Bacteria aren't something that you need to be afraid of necessarily. It's a normal part of your human health. It can help you with your digestion. It helps prevent you from getting sick from even worse bacteria. Overall, bacteria and other microbes can range from very bad to good. These are also a major part of the environment. The bacteria will really help us out a lot. These are going to be some of the things that help us get chemicals into our food supply that otherwise would be difficult to get in. For example, nitrogen is hard for us to break down and get into different plants, but bacteria can break it down from the air for us. These can also be great for commercial use. They can be used in biotech to research as well, but they can also cause diseases. So microbes are all around us. They are what we would call ubiquitous. That means that they're everywhere and you can't really escape them too well. You would have to do a lot to control your environment. So let's look at the different types of microbes. We have 2 major types of microbes that we're going to be looking at in this class. There's going to be the organisms and the acellular infectious agents. So organisms are going to be defined by having cells. The acellular agents are going to not have cells. They'll be acellular, as the name implies. So in the acellular infectious agents, we have 3 major types. We have prions, viroids, and viruses. Prions, they're going to be special, like zombie proteins. They're misfolded. They're basically dead and don't work. But what they're going to do is go and infect other proteins and cause them to misfold and become prions too. So this is an infectious agent that can be pretty dangerous. Viroids are going to be similar to viruses, but they lack a protein coat. They do have a small circular DNA, and they can hijack a cell and have it replicate the viroid. Viruses are also acellular. However, these ones will have a protein coat to protect their DNA or their RNA or whatever genetic material they use. Viruses are a little weird in that they can have odd combinations of genetics, but we're going to look at that in the future. But what you need to know about them is they infect cells and hijack the machinery of the cell in order to replicate. The other side, we have organisms, and we're going to look at bacteria, archaea, and eukarya. So our microorganism domains. When I'm talking about microorganisms, that means that they have a cell. So our 3 domains are going to be bacteria, archaea, and eukarya. Bacteria are going to be those true bacteria, the ones that you really know and you've learned about your whole life. These are going to be unicellular. They do not have membrane-bound organelles. The ribosomal RNA is unique to their group, and they have a peptidoglycan cell wall, and they're generally small. Archaea, these are not true bacteria. We'll go over them more, but they're unicellular as well. They do not have membrane-bound organelles. They do have unique RNA sequences. They do not have a peptidoglycan cell wall. We'll learn more about what that means later. And they're also very small. Eukarya, these are going to be unicellular or more multicellular. These do have membrane-bound organelle. They have unique RNA, ribosomal RNA. They do not have a peptidoglycan cell wall. Some can have other types of cell walls, but not all. And then their typical size is a little bigger than your typical bacteria or archaea. So when we go over some of these bacteria or other microbes in the future, we want to know kind of what their names come from. So when we look at E. coli, we see that the genus comes from the scientist that discovered it, and the species name comes from colon, the part of the body where it inhabits. Haemophilus influenzae, it comes from the word hemo, which is blood, and phile, meaning loving. And then influenzae is because we mistakenly thought it was caused by the bacterium that causes influenza. And then it just goes on like this. The names can either refer to who discovered it, what kinds of things it likes, where it can be found, misnomers where we thought it meant something else, or even other things like saccharose, meaning sugar, or mysis, fungus. So that would be sugar fungus. So just know that the binomial nomenclature, that means two names, will usually be describing the bacteria in some way. Like Staphylococcus aureus, Staphyloco means a bunch of grapes, so it kind of looks like a bunch of grapes. And coccus means berry, so the cells kind of look like berries. And we can see that more in the bacteria morphology. So we have two different ways that we're going to be looking at this morphology. We have the coccus, or we have the shapes of the individual cells, and then the shapes that the cells create when they reproduce. So let's start over here. On this side, A, this is going to be your coccus. This is your spherical cells. They're generally pretty round and look like a sphere. Your rods also are going to be called bacillus. These are cylindrical in shape. Vibrio is C, and this is a short curved cell, so it's kind of cylindrical, but it curves off. And then we have spirillum, which are very curvy. They're very spiralized. And then down here in E, we have spirochete, which are long spirals with unique motility abilities. Okay. Now over here, these different groupings are actually created because of how the cell divides. So if the cell is dividing in one plane, we call this binary fission with bacteria, by the way. So if a coccus divides, and it divides on one plane, one will go to the right, and the other will go to the left. And we can get a diplococcus. That means two cocci. So that means that you just have two of those spheres. Or we can get a chain of cocci. Okay, we can also get packets. So this is when we have two or more planes of perpendicular division. So it'll make these little cube shapes, possibly. And then we can also get clusters where it divides randomly in different planes. So this would be like Staphylococcus. And up here would be Streptococcus. So I said we would get back to archaea. Archaea is kind of special because it's not actually true bacteria. It has a different kind of cell wall, and we commonly call them extremophiles. That's because they can survive in extreme environments that most life can't. For example, if anyone's familiar with the Great Salt Lake in Utah, there's things called holophiles or salt lovers. These are types of archaea that can survive in the very salty water found there. You can also find some extremophiles in Yellowstone. These ones are called thermophiles. They love heat. So they're going to live in the hot geysers and hot pools in Yellowstone. Now, typically archaea are not going to be considered pathogenic because our bodies are not really conducive to them living. So they're not really going to be something that you have to worry about in your life too much. So eukarya are going to be cells that can or have membrane-bound organelles, and they can be unicellular or multicellular. So a few examples are fungi, algae, protozoa, and helminths. So we can also have even bigger ones. For example, humans are eukaryotic organisms. But since we're in micro, we're going to be looking at the organism studied by microbiologists. So fungi are going to be things like yeasts or molds and mushrooms. So they reproduce and they're fungi. Algae are typically going to be photosynthetic and use sunlight to produce energy. Protozoa are going to be single-celled microscopic organisms. They're going to use other materials for energy. So think like amoebas. And helminths are going to be like worms that infect your body. And their eggs and their larval form are microscopic. So once they get into your body, then they grow and invade the rest of your body. And then like we talked about, we have our acellular agents. That's going to be things like viruses, viroids, and prions or prions. So we've already talked about this. So that covers all of chapter 1. So now we're going to move on to chapter 2, the molecules of life. So life uses something called organic molecules. Oh, that can sound very special, but organic basically just means it's primarily made of carbon, as we can see here. We have a bunch of little C's, that means carbon. They're connected together by covalent bonds, and they can make 1, 2, or even 3 bonds with other atoms. These can either be linear bonds, as we see with these carbons, or they can branch off. And they can also make rings. So these are the different types we can see. Linear, branched, we have some called cross-link where they attach together, or networks, or rings even. So these organic molecules can help us make macromolecules. This just means a large molecule. Most of these macromolecules we're going to be calling polymers, and they're made of repeating subunits, also known as monomers. If we look at it, poly means many, mers would be the unit. Mono means one, mer is unit. So you have one unit or you have many. And we can see that over here, where we have our monomers being polymerized into a polymer. Polymerize just means to make a polymer. And we have 4 major classes of monomers. We have carbohydrates, we have proteins, lipids, and nucleic acids. So lipids are going to be kind of an odd one out, but typically a macromolecule just means that it has repeating subunits that are similar to each other. And so these sugars, amino acids, and carbohydrates are another name for sugar. These 3 are going to be the ones that have similar subunits that repeat. Lipids are a little bit different, and we'll cover that in a moment. So first, let's talk about how polymerization can occur. This is through a chemical reaction called dehydration synthesis. So it's also been called a condensation reaction, but they essentially mean the same thing. I prefer to use dehydration synthesis because once you break it down, it's easier to understand. So when we look at dehydration, if you know what it means to dehydrate, that means to not have water or remove water. And synthesis means to create. So when we use dehydration synthesis, we're removing a water to create a macromolecule. And we can really see that over here, where we have our 2 different molecules, and we do a reaction to make the water come out. So we remove a water and synthesize a polymer. So again, and then the other name, condensation reaction, it's because a water is condensing out. So pick either one and learn it, but know that both terms can be used interchangeably. The other term, the other thing we use when we're working with polymers and monomers is something called hydrolysis. So let's break it down. Hydro is referring to water, lysis is referring to cut. So hydrolysis is when we cut apart or break apart large molecules with water. So it's essentially just the same thing, but as dehydration synthesis, but acts in reverse. So we use a water molecule to cut apart a larger molecule. So this can either shorten a polymer into smaller polymers, or you can just isolate monomers and break them apart one at a time. So now that we've gone over some of the basics of organic chemistry and biochemistry, we're going to look at carbohydrates. And I'm going to keep it pretty general. I know this slide looks like overwhelming, but we're going to keep it pretty general. Carbohydrates are going to be made of carbon and water. That's because carbo means carbon and hydrate means water. So we have C, H, and O's. So there's going to be special ratio when we're looking at carbohydrates. Since it's 1 carbon with every water, that means our ratio is going to be 1 carbon, 2 hydrogens, and 1 oxygen. So carbohydrates are going to be energy sources for organisms and also a source of carbon, since that's primarily what carbohydrates are made out of. These can also combine with proteins to make something called a glycoprotein. These can be part of genetic material. We'll look at that when we look at nucleotides. These are also structural components of cell walls. We'll look at that later when we look at cell walls. And when we have one, it would be called a monosaccharide. If we have 2 carbohydrates together, it's a disaccharide. And if we have many more, it's called a polysaccharide. So some examples of some monomers or carbohydrates are glucose, mannose, galactose, and fructose. If it ends with ose, it's going to be a sugar. So we can go through and use that condensation reaction or dehydration synthesis in order to combine those different carbohydrates together. So we'll remove a water and create a larger molecule. In this case, it would be a disaccharide, sucrose, which would be the combination of glucose and fructose. And we can even grow these longer and build different things like cellulose. That's going to help plants make their cell walls, or glycogen, and that is going to be how humans store sugar for use later. The next thing is going to be a lipid. Lipids are going to be hydrophobic molecules. So hydro means water, and phobic means afraid of. So these are water-filling. This means that lipids are going to repel water. The monomers are typically going to be glycerol and fatty acids. Now, something special is that glycerol is actually a small little tiny type of sugar, and fatty acids are going to be just these long chains of hydrocarbons, or hydrogens and carbons. So the crazy thing here is these are actually very different molecules, but we still class them as a macromolecule because they function similar to one. So lipids are going to be mostly made of carbons and hydrogens, and they're going to have oxygens at the end of those fatty acid chains. So we have a diverse cast of lipids. They're going to do different things. So triglycerides are going to be the primary energy storage for long term. Phospholipids are going to be a type of lipid that makes up the cell membrane. Steroids are going to be the special four-ringed molecules that can create hormones, but steroids can also be converted into cholesterol, and that can help your cell membrane have the proper rigidity or fluidity so it can move around properly. The next macromolecule are proteins. Proteins are going to be a macromolecule made up of amino acids. Now there are 20 different types of amino acids that humans will use. So amino acids can combine by making a peptide bond with the amino group of one molecule and the carboxylic acid or carboxyl group of another. And this will combine and make a long linear chain. So it makes a straight line. So the functions of the functions of proteins is going to be really, if there's anything your body's actively doing, it's probably a protein. So enzymes are going to speed up chemical reactions in your body. We call these biological catalysts. These can act as transporters, so they can allow things through the cell membrane of a cell, or it can even push something out of a cell. Signal receptors, these things are how organisms receive signals from each other. Motility allows the protein or the organism to move, and support is going to just create the structure for a cell. So the proteins rely on folding for their function, and folding is affected by various different factors, including the pH, that's acidity, the water content, the heat in the area, or chemicals, also called solvents, or solutes. So the acidity can cause, any of these things, if they're out of the ideal conditions, can cause a protein to denature. That means that the protein is going to unfold and not work. So what this folding looks like is we have many different structures. We have a primary structure, and that's just the chain of amino acids. We have secondary structures, and that's just going to be a general folding. And those will contribute to tertiary structures. That's just a larger group of secondary structures. And this is going to start being 3D, and it's hard to demonstrate on this screen. At the tertiary structure, proteins can actually start to do stuff. Now, if you take many tertiary structures and add them together, you can get a quaternary structure. So the quaternary structure will be made up of independent tertiary structures, meaning that they're not actually connected, but they do interact with each other in special ways. Nucleic acids are going to be the macromolecule that is in charge of your genetic material. So these are going to be things like DNA and RNA. So the subunit of nucleic acids are called nucleotides. Nucleotides have their own subunits. So we have a ribose sugar, or sometimes a deoxyribose in the case of DNA, a nitrogenous base, and a phosphate group. So when I said that sugars will be involved in DNA, it's because it is this part, the deoxyribose sugar in DNA. So we actually have a few different types of nitrogenous bases. We have adenine and guanine, those are your purines. And then in the pyrimidines, we have thiamine, cytosine, and uracil. Now, thiamine is only found in DNA, and uracil is only found in RNA. But the other three are found in both. So when you start to polymerize this, you can start to build a long chain of DNA. So you add nucleotides on at the 3' end. So the side with the phosphate is going to be the 5', and the side where we can add on is the 3'. And that just gets the name, or the 3' and 5' just gets its name based on what number carbon it is away from the oxygen, starting on the right. So 1', 2', 3'. So this would be our 3', and that's where we can add another nucleotide onto. The 4', 5', this is where our phosphate is. This is where it started. So we can add on to the 3'. So when we have two strands of DNA, we call these complementary strands, they can begin to form hydrogen bonds with one another. Now there's a special bonding pattern that you can see here. Adenines will bind with thiamines, they make 2 hydrogen bonds with each other, and cytosine will bind with guanine, they make 3 hydrogen bonds. In RNA, uracil would take the place of thiamine, and so uracil would bond with adenine. Now we have one major place where we find DNA, and that's going to be in the nucleus or the nucleoid. But you can find extranuclear DNA sources like mitochondrial DNA. In humans, that's DNA found in the mitochondria. And we also have plasmids. These are small chains of DNA in things like bacteria. So those two are going to be extracellular DNA, or extranuclear DNA. They're in the cell. That covers all of Chapter 2. So now we're going to move on to Chapter 3. These are going to be the cells and the methods to observe them. So when we're looking at cells, one of the biggest ways that we determine cells is based off of its cell wall. Now the cell wall of a bacteria is going to be primarily made of a substance called peptidoglycan. So peptidoglycan is a strong mesh-like network only in the bacteria. So only bacteria demonstrate this specific cell wall. And as you can see here on the right, we have examples of this. And this is only a 2D image. And you see how mesh-like that is. And if we have it, if we were to look at it in 3D, we would see it has a lot of layers that protect the cell. So bacteria cells will use a special form of amino acids called D-form amino acids. Humans will typically use a form of amino acid called L-forms. The way that I remember the difference for the amino acids is humans want to live, so they use L-forms. Bacteria cause death, so they have D-forms. You also have carbohydrates in there. So the carbohydrates that make up peptidoglycan are going to be alternating N-acetylglucosamine and N-acetylmuramanic acid. Or, because that's a mouthful, NAG and NAM, N-A-G, N-A-M. And we can see that here. We have NAG, NAM, NAG and NAM alternating. And then we have a tetrapeptide chain of amino acids that creates a cross-link between two different chains of NAG and NAM. So this is where that peptide part of peptidoglycan comes in. Peptide means protein. Glycan means sugar or glucose, sugar. So when we look at this tetrapeptide chain, we can see something unique. We have a interbridge or a cross bridge in gram-positive cells. Now, this is actually going to make this amino acid very special because it's not just making a long chain like we had figured out amino acids do. It's making a branch. Typically, amino acids don't make a branch. So this means that bacteria are going to have a special amino acid that's capable of making 3 peptide bonds instead of 2. Now this is called diaminopimelic acid. It's a mouthful, but that's what it's called because it can make 3 peptide bonds. So your peptidoglycan and the cell wall is going to prevent bacteria from bursting because it gives it a strong, rigid outside, and it gives the bacteria its shape. Well, if it keeps the bacteria from bursting, and it's unique to bacteria, that gives us a target to attack in order to kill bacteria to keep people alive. But we'll cover that more later. Let's look at the first type of bacteria. This is called a gram-positive bacteria. We call these things gram-positive because they are positive on the gram stain. This means they show up purple. We'll cover that later on more in detail. So your gram-positive cell walls are going to be typically thicker than your gram-negative cell walls. They're going to have up to about 30 layers. Now, they do have gaps in them, so small substances can pass through and reach the cell membrane. Now, something special about gram-positive cells is that they have something called teichoic acid that passes through the cell wall, and this accounts for about 60% of the mass of the cell wall. So when we look at this image, these long red chains are called teichoic acid. The ones that are passing through the peptidoglycan, that portion of the teichoic acid is called walled teichoic acid, and this is going to attach and bind to the peptidoglycan. The part that goes down and attaches to the cytoplasmic membrane or the cell membrane is called lipoteichoic acid, because it's attaching there. Lipo for the lipid, the phospholipids that it's attaching to. So teichoic acid can have many different functions. The one that is most common is that it binds cations like magnesium. It may have other types of uses, and those are actively being researched. Next, we move on to gram-negative cell walls. These are going to be red or pink on a gram stain. These will typically have a much thinner layer of peptidoglycan. However, unlike gram-positive, gram-negative will have an outer membrane. So this outer membrane is going to be a lipid bilayer with proteins on it, and it's attached by lipoproteins. Okay. And we're going to look more at this in the next slide. So the outer membrane is going to be made of lipopolysaccharides called LPS instead of phospholipids like the lower membrane. So LPS will, when it's injected into animals or humans, it can cause fever and other symptoms. Now these symptoms are going to be the body's natural response to LPS. This is a normal indication that gram-negative bacteria has invaded the animal. However, in large quantities, LPS can be problematic. It can cause shock. This can lead to a drop in your blood pressure and be a life-threatening event. So we call LPS an endotoxin. This means that it's toxic when it gets inside of you. LPS is made up of two parts. So when we look here, we have lipopolysaccharide, LPS, and there are two parts. We have lipid A, and this is going to be the part that makes the bilayer, and we have O antigen, and that faces outside of the cell. So that O antigen is the part that we are detecting. And different antigens or different bacteria will have different types of O antigens. So that's how we can identify different types of bacteria. So your outer membrane is mainly going to function to help protect the cell and its cell wall. So this is going to prevent things like antibiotics or saliva from getting in there to attack the peptidoglycan, but more on that later. Your outer membrane has things called porins that allow small molecules to pass through in order for the cell to get its food. So now let's look at some antibacterial substances because I've been alluding to it. So these substances that we're going to talk about right now are mostly targeting the peptidoglycan, that special cell wall in bacteria. So let's look at our antibiotic drugs, penicillin. So penicillin will actually interfere with peptidoglycan synthesis. So it'll prevent peptidoglycan from being produced. So this means that growing gram-negative cells are more at risk than most other growing gram-positive cells are most at risk. Gram-positive cells are typically going to be more affected by penicillin because gram-negative cells' outer membrane prevents most of the penicillin from entering. So gram-negative is naturally a little protected from penicillin or other drugs of that class. Lysozymes are going to be an enzyme, a protein found in tears, saliva, and other bodily fluids. This destroys the structural integrity of peptidoglycan. So this means that gram-negative cells' outer membrane prevent the lysozyme from entering, meaning this is more effective against gram-positive. So in both of these cases, being gram-positive makes them more susceptible to damage to their cell wall. And here's just a brief rundown of what's going on here. We have gram-positive. They're going to show up purple in the gram stain so you can see them. They show up purple. They have thick layer of peptidoglycan. They have teichoic acid. They do not have an outer membrane, so they do not have lipopolysaccharide. And they do not have porine proteins. So they're generally more susceptible to penicillin, and they're more sensitive to lysozyme. On the other hand, gram-negative is going to show up pink on this gram stain, and this is going to have a thin layer of peptidoglycan as detailed on this. It will have an outer membrane, it will have lipopolysaccharide, and it will have porine proteins. This is generally less susceptible to penicillin. There's some exceptions, and it generally is not sensitive to lysozymes. Other structures on the outside of the cell are things called capsules and slime layers. These are gel-like outer layers. So they're made up of polysaccharides. Something special about polysaccharides is that they can attract a lot of water, so that's what makes it gel-like. We will often call these capsules or slime layers glycocalyces, or a glycocalyx if we're talking about one. These little slime layers, these capsules, can help form biofilms. Now, biofilms allow these bacteria to attach to places they normally couldn't. One example is on your teeth. The plaque you have is typically a biofilm that allows bacteria to survive there. It also helps protect the bacteria from your body's immune system. So it prevents phagocytosis of the bacteria cell, so they can't be eaten by your body's defenses. Another structure on the outside of the cell is a flagella. This is a long protein structure. This enables movement of the cell, and we can see that here over here. If we have multiple flagella, this is peritrichous, and if we have a single flagella, this is polar monotrichous. So polar because it's on one pole of the cell, and monotrichous because there's one, or peritrichous because there's many. So your flagella will have three major structures. You have your basal body. This is the part inside of the cell membrane. You have the hook that attaches the basal body to the filament. That's the long whip-like structure that goes out. Now, this structure does not actually look this mechanical. This is just a representation in the book. So cells that are able to move will typically have the ability called chemotaxis. This means that they move towards a chemical that they detect. So if these bacteria send something, it'll make their flagella start whipping and they'll move towards it. The next structure we're going to look at on the outside of a bacteria is something called a pili or pilus. This is much shorter and thinner than a flagella, and this is a protein with a hollow core. Now we have a few different types of pili. So the first one is a fimbriae. This allows it to attach to surfaces. So one example is if a bacteria is giving you diarrhea, that bacteria does not want to be rushed out with the waste. So the fimbriae help the bacteria attach to your intestinal linings. Some pili can give it mobility when they're on solid objects. And the last type of pili is a sex pilus. This transfers DNA from one bacteria to another. This can help share important genes that can keep the bacteria safe in the future and thrive. Inside of the cell, inside of bacteria cells, we have the DNA, and in this case, we're looking at chromosomes and plasmids. So chromosomes are going to be the main DNA of your bacteria. These are circular double-stranded DNA. These are called haploids. Bacteria are called haploids because they only have one copy of DNA. We call this chromosome a nucleoid. Bacteria have nucleoid. And then plasmids are smaller and similar to chromosomes. These are independent from the chromosome, though. They're not a part of the nucleoid, and they have additional genes that can be transferred via the sex pilus. So these are genes that can give antibiotic resistance, for example. Ribosomes are another structure inside of the bacteria. These are made up of two smaller subunits, and these facilitate peptide bonds. This means they facilitate in building proteins. They're different from human ribosomes. Human ribosomes are called 80S ribosomes, whereas bacteria are 70S. This makes the bacterial ribosome another target for antibiotics to attack. Something to keep in mind about antibiotics is that we always want them to be able to attack something that bacteria have and we don't. So in this case, it would be the unique type of ribosome. And some other feature that not all cells have is called an endospore. So if an endospore is a dormant state, and this can be produced by endospore-producing cells. So if we look at this example, we have a cell, and it chooses to stop growing due to some environmental factor. And what it does is it replicates its DNA, and then it walls it off, and it creates this little endospore which can survive. Now, when conditions really get bad, then the mother cell will die, and the spore will escape. And this is going to be a dormant cell waiting for conditions to improve. And once conditions can improve, the endospore can germinate again and become a vegetative cell. This is a cell that is actively living. Now, if you notice, in this example, we create an endospore, but the mother cell dies before the endospore is released. This means this is not a form of reproduction. This is more like an escape hatch. So the cell can survive even though if it was vegetative, it would die. This makes it so endospores can survive extreme conditions like high heat or lack of water. Other structures found within cells are things like the cytoskeleton. This contributes a framework to the inside of a cell, and it can help with cell division. Remember, in bacteria, we call that binary fission because it creates two cells and it divides. Storage granules are going to be different density substances that just collect in one area. So imagine like a dust bunny collecting in the side of your room. That's similar to a storage granule. Protein-based compartments are going to be small, rigid structures that can isolate things for the cell. Now do note that bacteria cells are not as specialized as eukaryotic cells, so they don't actually have membrane-enclosed organelles, so don't confuse protein-based compartments with organelles. They are not the same thing. All right, well, that's this whole lesson. I was not able to cover everything in the book or in the reading, so please do not forget to go back and read. Have a good day.